Wake up, hello and welcome to another video presentation with Victor Black. In today's video we're going to be talking about steroid esters. Steroid esters are one of many subjects in this space that are widely misunderstood and widely confused by the community. So the purpose of this video is to act in the first instance as a primer, an introduction to the concept of steroids, esters, what their role is, how they work, and then undoubtedly there will be follow-on videos. I'm sure guys are going to have questions that follow on from this. I already have things I want to talk about separate to this, but it's important in my opinion that we start with a 101, okay? A high-level discussion about what esters are, what's their role, and what do you need to know about esters to use these compounds effectively. Okay, so let me begin by explaining to you in the simplest way possible. As always, I see my role here as simplifying the information to the point whereby we can say, well, what can we do with that, right? It's possible for us to have a conversation that is, in my opinion, very, very, very technical about how all this stuff sort of gets put together, and the reality is as always, I'm interested in, so what can we do with it? What we have this knowledge, how do we apply it to get an outcome, right? So the focus of this video is going to be relatively simple conversation about esters, and then so what does that mean to us? What can we do with that? All right, how can we apply that? All right, so let's start off by understanding that everything that I talk about here today, I'm going to kind of reference a couple of compounds. I'll talk about testosterone, I'll talk about nandrolone, but understand that these principles are universally applicable to all esterified steroids. So if there's an ester attached to a hormone, this framework that I'm talking about today applies to that. Okay, so what's an ester about? Fundamentally, the reason that esters developed, and they're not exclusive to the world of steroids, they're used in many different medications. The purpose of attaching an ester to a compound is to control in an efficient manner its release into the bloodstream. Okay, so we need to deposit the compound into our muscles, as it were, through an intramuscular injection, and we're going to talk about subcutaneous as well, but predominantly through an intramuscular injection. And then that compound is going to go into our bloodstream, and effectively then there will be a sequence of events that occurs downstream from that. In the case of steroids, all right, the steroid hormone basically gets into the bloodstream and then ultimately passes into a cell, and at a cellular level it binds to a androgen receptor that in turn triggers gene transcription, and that in turn translates into proteins being produced that alter the cell activity, right? So that's the high level concept of what's going on here. The hormone gets into the bloodstream up here and then ends up in the cell and does its business. We're going to talk about this in a separate video, right? But the problem with getting it into our bloodstream is we don't want to just deliver it all at once, like in a bolus dose. Here, have all this testosterone, right? And that fundamentally is what would occur if we were to introduce testosterone as a standalone hormone, right? And this product is available. This is basically what's known as testosterone suspension. It's simply the testosterone hormone suspended in aqueous solution, and you administer it, and immediately basically is then available for use at the androgen receptor. The challenge with that is, remember, these drugs are not developed for bodybuilders. These drugs are developed for on-label medical use. The challenge with that is you don't want to be injecting this stuff every single day. Yes, I do that, but the average person doesn't want to be injecting testosterone every day. So the solution to this is to place an ester against the hormone. And there are a significant number of esters that are used in steroid hormones. They range from things that are well-known products like propionate and enanthate. There's on and on and on and on and on the different esters you hear about. There's probably something like a dozen different esters, right? The esters that you're looking at on this page, this here, right? These are the 4 esters that in combination make up the product Sustanon. Most people have heard of Sustanon. It's a multi-ester testosterone product. So it has some propionate and it has some of the different esters in there. And these esters range from quite fast acting to quite slow acting, all right? So basically what this is all about is about timing, about delivery of the steroid hormone into the bloodstream and how we can regulate that process. Okay. So before we begin that discussion, I want to basically step back one step, and explain to you the bigger picture here, because this is the part that most guys miss. All right. There's a number of things that actually impact on the timing or the delivery of these compounds into the bloodstream. It's not just esters. So if the purpose of esters is timing, it makes sense rationally and logically to step back and say, well, is there anything else in this space that we should be discussing at the same time? And the answer is absolutely. Very rarely do you hear this conversation come up when people talk about esters. In fact, almost never. But it's important to understand that you can have as profound a difference between two different esters, right, through the administration practices as there is between the esters themselves. Let me explain what I mean by that. There's several factors that will influence the delivery of the steroid hormone into the bloodstream. The first of which is the chosen injection method. So we know that we can do this intramuscularly, we can also do this subcutaneously, and both of these methods have different timing deliveries attached to them. Okay, so if you inject intramuscularly, if you inject subcutaneously, both of these injection methods have different timings associated with them. Subcutaneously is much slower for the hormone to reach into the bloodstream as intramuscular is. Quite profoundly so. There's at least as much of a difference, in my opinion, between these two methods as there are between two esters, which is interesting when you think about it. Okay. The second thing that a lot of guys are not aware of is the injection site has a profound impact on timing and release of these compounds. If you inject into your glutes or your shoulders or your thighs, each of these locations have different timings attached to them. All right. The injection volume changes the release of these compounds. So the larger the deposit of oil, the more slowly that the deposit is absorbed, and therefore the steroid hormone is released. Okay, the carrier oil, the oil that's actually used to suspend the hormone, there's many different types of carrier oils, and different carrier oils themselves can actually influence the timing or the release of these steroid hormones, right? And there's always going to be a degree of genetic variation, right? So each individual has an individual consequence, all right, based on their genetic makeup of the release of these compounds, right? So the reason I'm saying all this is you need to understand that if the discussion we're actually having is about release of the compound from the point of administration into the bloodstream, then you're in these other subjects, which are very rarely discussed, should be discussed on the same page as esters, because they do the same thing. Esters are about timing control. These other things are about timing control. They have an impact, so we need to discuss them all at once. All right? The other thing I just mentioned in passing here, although I've never found any evidence to support this, there's pretty good evidence that when it comes to water-based injections, things like HGH and insulin, those sorts of things, exercise can actually impact on the absorption rate of these drugs from administration points. So it's possible that concept also applies to oil-based deposits. All right. So my point is, this conversation suddenly became much wider than the concept of just an ester. We need to now talk about, well, where are we going to pin this material, right? How much are we going to use? What method are we going to use? You're going to do IM, are we going to do SC? All of these things have the potential to have a very profound effect on the time it takes for the steroid hormone to basically be released from the injection site and then pass into the bloodstream and then go through the process of being ultimately available for the androgen receptor. Okay. So let's just quickly talk about that concept. So here we have an injection of an intramuscular steroid into the muscle itself, and oil deposits made. All right. And then over time, basically that oil is basically released into the bloodstream a little bit at a time. Doesn't happen woof like that. Like, there's not immediate release, right? And you can see here they're quoting times of 8 to 14 days after administration, and we're still seeing oil being released, right? Now, what that looks like, if you've never seen this, I posted something on this today. This is an MRI scan of an intramuscular oil deposit. All right, so this is basically what it looks like on an MRI scan. And you can interestingly see all these tiny little dots. And hopefully you can make that in the video. There's all these tiny little dots, and basically what they are they're little bits of oil that have broken away from the main body of the thing. Now, the reason I point that out, it's important to understand that the oil in this shot is absorbed into the body slowly over time, right? It takes several days for that to happen, right? And it's the little kind of spots, the breakaway kind of things here that actually are absorbed most quickly. The larger the surface area of the deposit, the more slowly that the oil's picked up. So what that looks like over time is this. This is basically the same deposit, right? And as you can see over time, as the oil is absorbed and taken up, the surface area gets greater and greater and greater relative to the oil content itself. And so it speeds up, the process speeds up, right, over time. The more these little kind of fragmented pockets of oil are, the faster it can pick up the oil and administer it. So the delivery of oil is not a linear thing. It happens in a non-linear manner. Okay. And this is just for interest, if anyone's interested. This is what a subcutaneous deposit looks like. So you can see it's a different shape than the oil administration done with the intramuscular, right? Now that is interesting because the different shape means that you'll get a different surface area, and a different surface area means a different delivery time of the compound. Okay, so where are we here? Okay, so basically what then happens is over time, gradually, small amounts of the oil deposit are absorbed by the body, taken up and dispersed by the body. And when the oil is absorbed, the steroid hormone attached to its ester is then free to enter into the bloodstream. And when that happens, not before, when it enters the bloodstream, it then interacts with enzymes that effectively remove the ester. It's called cleaving. So basically what happens is there's an ester that's attached to this, and I'll explain this in another video about structural modifications of hormones, but there's an attachment point where they physically attach an ester here. Right? If I go back to here, it's just here. Okay, so this is where they attach the ester, right? And what happens when we cleave it is basically the enzymes come along. It's a natural biological process. Esters are not specifically connected just to steroids, and it physically removes the ester, right? And what happens when that occurs, right, is that the principal hormone is then made available so that it can bind with the androgen receptor. It can then make its way through the bloodstream, usually bound to proteins, in such a way that ultimately ends up inside the cell and binds with the androgen receptors. But what's important in that process is to understand that enzymes need to come along and cleave this connection. Get esters. Fundamentally, in the simplest way possible, what we're saying here is that each one of these esters has a varying degree of complexity for it to be cleaved, right? And the degree of complexity means that it takes different time frames. So something like propionate, which is, everybody knows, a relatively fast ester, is very simple in the scheme of things for enzymes to cleave the ester and then allow the testosterone hormone molecule to go on its way and do its thing, right? Whereas the DECA, testosterone DECA, basically is more complicated, and therefore it takes longer to do that. So this is a longer ester and it's longer acting. Just out of curiosity too, so if anyone's unaware of this, a lot of people refer to nandrolone as DECA. DECA itself is the ester that's attached to nandrolone. The hormone is nandrolone. For some reason, I don't really understand the historical context, but it became known as the ester, right? In the same way that propionate, sometimes known as propionate, but the actual hormone itself is testosterone. The common hormone through here is testosterone. These are the esters. This is what is removed to leave behind the testosterone molecule, the testosterone hormone. And what's important for guys to understand is that when the steroid hormone with the ester attached goes into your bloodstream and the ester is cleaved and the ester is cut off and removed, right? What is left behind is the testosterone hormone, just looking exactly like it would if it was in suspension, okay? So what confuses people sometimes, they go, how are these products different? They're all the same. They're all exactly the same molecule. These are all testosterone, all testosterone, all right? They're not different. The esters are different. So once we cleave the ester, once it happens, and then the testosterone hormone is free to go and bind with the androgen receptor, they're all the same. There is no difference, okay? There are some fundamental differences in how much active ingredient is in, say, for example, a milliliter of compound. The reason behind that is each of these esters has a different molecular weight, right? So that weight needs to come out of the dosings, as it were. So, for example, the molecular weight of DECA as an ester, right, is heavier than it is for propionate. So there's more testosterone in a milliliter of test propionate than there is in a milliliter of decanate, because you need to remove the molecular weight of the ester. If that's confusing, I can explain that. You know, anyone that's confused about it, let me know. I'll try and do a better job of explaining that. Okay. So basically, as I said, in another video, I'm going to explain how structural modifications of hormones happen, testosterone. There are basically these 4 rings, okay? And at position 17 on the D ring here, we basically attach an ester. We cleave that off to free the hormone to go and do its thing, testosterone hormone to go and do it. And again, if it's a nandrolone hormone or whatever, we cleave the ester off, the hormone is then free to go and do its thing. So there's a couple of things that confuse people here, the big things. One is people don't understand that, you know, like at the end of the day, testosterone is testosterone. Once the ester's cleaved, it's the same hormone. It's exactly the same hormone. People will swear black and blue that they feel that they get different results from different esters. And they go, you know what, there has to be more to this conversation than that, because if I use, you know, such and such an ester, I feel different than I use the other ester. So how can it just be testosterone? You know, I feel different. Let me take a few minutes to explain how this works and clear this up for people, right? And to use this, I'm going to use the most profound example that I can use, which is in the nandrolone esters, right? So when you talk to people about nandrolone, they will swear black and blue that NPP, right, and decagerabulin are different compounds. Okay, they're different. I use them and I feel different when I'm using them. Let me explain what's going on there. Do they feel different? Yes. It's a very credible thing to do, but you have to understand why, right? If you take a product, right, that has 100 milligrams, it's 100 milligrams. This chart that we're looking at, by the way, is the administration of 100 milligrams of nandrolone, okay? And you release that 100 milligrams over a time frame of, let's say, 5 days, right, in its totality, right? That's not exactly what's happening here, but bear with me, right? Then you're going to see a peak value of a certain amount of compound, right? If you take that same 100 milligrams of product and release it over, let's say, 30 days, right, same quantity of product, you're going to see a much lower peak of product, right, peak value in serum levels, much lower, right? And that's fundamentally what you're looking at here. So this is the peak value of nandrolone, this is the peak value of DECA, all right? Why is it different? Right? The reason is they're both 100 milligrams. So why the much higher peak? It's quite simply because nandrolone starts working here with the NPP, right? And for all intents and purposes, let's just arbitrarily say by the time it gets here, it's run its course, right? So we've got 15 days of a reasonably significant presence, right, of this product, right, that produces a peak value all right, and a duration. That's the consequence of a 100 milligram injection, right? Now you can see when it comes to DECA, same compound, right, same nandrolone compound, but a different ester. The peak value is much lower, but the duration of exposure is significantly greater. So we're out to here, we're at 30 days here for a single 100 ml shot. And realistically, we still haven't run our course yet. Anyone that's familiar with, DECA law, bro law, you'll understand this is a very, very, very long acting ester, right? So what matters, if we extend this out to, let's say, 60 days, it's actually probably longer than that, right? What you need to add up is the total time under the curve, right? So you have this time here, okay, all of this time, right, out to 60 days, all right? And then what you have on the other compound is a much higher peak, but it basically drops down, and let's say at 15. So the total amount of exposure to the compound is a result of the 100 milligrams, but it's about how quickly it was pushed into the bloodstream to become active. Let me show you something that almost everyone's familiar with, and it might make a little bit more sense. This is what sustanon looks like when 4 different esters are released over a time frame. So this is the administration point for sustanon, okay? You can see that propionate, the fastest acting ester, right, peaks very, very quickly and it has a high spike, and then it's all over in let's just arbitrarily say 5 days. It's kind of done, a little bit of dribble-on effect, right? So it's a very high peak value, but not a very long duration. In the same time, the longest ester here, DECA, has a very, very low peak value compared to propionate, but it's still going on. It's off the paper here, okay? So in sustanon, these are not all identical volume of compound, right? So just bear with the explanation here. But the idea is that if you have an ester that acts very quickly and releases its load into the bloodstream very rapidly, all of that active compound becomes available in a short time frame. So the peak value of the hormone is very high. And what that means is the performance outcomes are significantly higher. Significantly higher. The peak value here and the peak value here, you're gonna get different outcomes from this compound. Right? It's the testosterone here and the testosterone here are going to impact you differently, not only in good ways, but in bad ways as well. So if the testosterone peak is very high on a short ester, then so are the consequences of side effects. So is the aromatization in the estrogen. So is the issue with conversion into DHT. If you keep a lower peak, right, and you take it over a longer time, potentially you're going to have less performance and less side effects from what amounts to the same amount of the hormone. There's no difference between the same amount of hormone. The idea that you can take a hormone, cleave its ester, right, and then introduce 100 ml, as you would in something like nandrolone, and 100 ml, like you would in, say, NPP. So this is DECA, and this is NPP, or this is, test propionate, and this is test DECA, and that you're going to get a different result from 100 milligrams of the same hormone, doesn't make any sense. It's completely implausible. But if you release that 100 mil in 3 days, or you release it over, in 30 days, then it makes perfect sense what's going on. Of course it feels different. It's almost a completely different experience, but the underlying biology here is that it's the same amount of the same hormone being released. Right? A lot of guys just, they can't make that connection. It's the same compound. It's just released faster, and because it's released faster, it produces a high peak value versus a longer duration. And the consequence of that is it can feel very, very different. It can feel like a completely different drug. Right? Different performance outcomes, different side effects, but it's the same hormone, guys. Okay, so fundamentally, that's really where I want to kind of leave a primer. There's a dozen questions that we can break away now and talk about esters, at this and this and this and this, but again, you have to make a primer a certain length. So hopefully, as a primer, that makes sense. Let me just recap quickly. Esters are all about timing the release of the steroid hormone into the bloodstream in an active format. So basically the oil deposit then gradually breaks down. It slowly drip feeds as it will in a non-linear fashion esterified steroids into the bloodstream. All right, doesn't all come at once. Drip, 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 non-linear. Once it hits the bloodstream, the enzymes in your blood, there are enzymes in your blood that are able to cleave off the ester, to get rid of the ester. Okay, how long that takes is a result of what ester you're using. The more complex the ester, the longer the chain, the longer that process takes. But once the ester has been cleaved, once it's been hydrolyzed and cleaved, and it's free to go and basically interact with the androgen receptor, that hormone is that hormone. That's it. All right. Testosterone is testosterone. Nandrolone is nandrolone. That's basically it. All right. So hopefully that's a good high level 101, guys. I'm sure you've got questions. Please send them in and I'll do my very best to answer them. I hope you enjoyed today's Victor Black Masterclass video. Sawadee Cup.